acids and glycerol, free fatty acids bind to albumin because they are insoluble within the aqueous environment of plasma. Now, let's zoom in on a hepatocyte where a fatty acid has entered the cytosol, and then further to its mitochondria. Mitochondria have a relatively porous outer membrane and a relatively impermeable inner membrane. Between these two membranes is an intermembrane space, while the matrix is the larger space surrounded by the inner membrane. Let's take a closer look. Acyl-CoA synthetase and carnitine acyltransferase 1 are embedded in the outer membrane. Carnitine is within the intermembrane space. Finally, acylcarnitine translocase and carnitine acyltransferase 2 are located in the inner membrane. When a fatty acid is delivered to a cell, it must be transferred inside the mitochondrial matrix in order for its oxidation to take place. A fatty acid cannot enter the mitochondrial matrix by itself, so an elaborate shuttle system is used to facilitate its entry. A fatty acid binds to the enzyme acyl-CoA synthetase on the outer membrane, which uses ATP and coenzyme A as substrates to produce acyl-CoA. Acyl-CoA then crosses the outer membrane to the intermembrane space. Here, carnitine acyltransferase 1, located on the outer mitochondrial membrane, facilitates the exchange of carnitine with the coenzyme A of acyl-CoA, thus yielding acylcarnitine and coenzyme A. In order for acylcarnitine to cross the inner membrane into the matrix, it must pass through an antiporter called acylcarnitine translocase. This antiporter will simultaneously exchange the acylcarnitine in the intermembrane space with a carnitine from the matrix. Once acylcarnitine is inside the matrix, carnitine acyltransferase 2, located on the inner leaflet of the inner mitochondrial membrane, catalyzes a reaction that transfers the fatty acid from acylcarnitine onto coenzyme A. Thus, acyl-CoA is reformed and carnitine is liberated. The net effect of the carnitine shuttle is that the acyl-CoA that was on the outside of the matrix is now located within the matrix. Inside the mitochondrial matrix, acyl-CoA undergoes many cycles of oxidation through the action of a fatty acid oxidase complex. In each cycle, there are enzymes, such as long-chain and medium-chain acyl-CoA dehydrogenases, that oxidize the fatty acid by removing high-energy electrons, transferring them to FAD to form FADH2, and to NAD to form NADH. The FADH2 and NADH formed in each cycle will generate 5 ATPs per cycle using the electron transport chain, or ETC. In addition, each cycle will split off the 2-carbon acetyl-CoAs. As a result, the fatty acyl-CoA is shortened by 2 carbons in each cycle. The fatty acid will go through continuous cycles of oxidation until broken down solely into acetyl-CoAs. The number of cycles needed for fatty acid oxidation can be calculated by dividing the number of carbons in the fatty acid in half and then subtracting 1. For example, the oxidation of palmitic acid, which has 16 carbons, undergoes 7 cycles of oxidation.